So we're getting close to the end of the semester. I'm sure most of you are happy about that. So if you had been looking ahead, you saw that I had some mistakes in the syllabus because I had some double dating back there. So this is where we are right now, 419, and we're finishing up this lecture on sedimentation. We'll meet Friday again. I have a 10:30 meeting. I think I have a 10:30 meeting Friday. Oh, it's at 10 to 12. So one possibility is to do it at 12. Or a little bit after 12, but you all have to sort of agree. So is there any problem with 12 to 1 on Friday to go over another set of assignments?
I'm good with 12.
Anybody who has a problem? Nobody? Okay, so we're going to meet at 12 o'clock on Friday. Save that, and I'll send you. Oh, I'm sorry, I wasn't done talking to you. Next week we're going to do some papers, and this will involve a merging of sedimentation analysis and some early methods that people use for analyzing sedimentation data, and then I'll cover a related paper that we published that uses quote unquote more modern methods. And the first two papers, there's a lot of thermodynamics and a lot of thermodynamic linkage in it. So we'll talk about that. And then the week after, which I guess is the 3rd, we're going to do microtubule assembly. And again, an older paper that merges thermodynamics with macromolecular assembly. And then a paper that my lab published that merges some of that and gives you a more modern perspective on it. We combined a lot of data from the entire field at that time. Okay, originally I thought I was going to cover another sedimentation set of papers on zinc alkaloids and tubulin, but we're running out of time. And so I think what we need to do is cover this paper from John Ladbury's group that looks at the use of thermodynamics in drug design. And so we'll discuss that paper. This will be a nice summary to a lot of the thermodynamics that we've discussed. And then we'll have 2 weeks to do student presentations, okay? And I'm almost ready to give you the papers for that. I'm still searching around for something that I like better than what I have. Okay, so that's sort of where we are. The assignment that you're going to get, it's already been posted out there. From here on out, we'll be involved in discussion. And so I think if I make assignments, is this all written as New Times Roman? Okay. We'll mostly be discussing, and this final assignment here, this final discussion here on the 10th will be a great summary for the 4 of you who are taking the prelim. Okay. All right, so let's begin. And I'm pretty sure, I didn't go back and double check. I'm pretty sure we had finished sort of here with density, viscosity, and I mentioned that we have a density meter and a viscometer. We've actually got 2 viscometers last time. Okay. Hello, move. Okay. So sedimentation velocity is also referred to as moving boundary. And one of the things that comes up, we've spent a lot of time talking about the frictional coefficient and Stokes-Einstein, and turning this into information about shape, assuming spheres. And we talked a little bit about the Perrin approximations for oblates, prolates, rods, and the production of an equivalent sphere. So if this structure were a sphere of the same volume, how big would the radius be? And the value that you end up calculating usually is called F/F0. Okay, and so you can see in this equation that there's an F/F0, and it involves a concentration dependent term. So there's a concentration dependence to the frictional coefficient. And what this does, because the diffusion coefficient and the sedimentation coefficient both have a frictional coefficient in the math, what this does is it adds a concentration dependence to the sedimentation coefficient. And so you can see here the form of the equation involves 1 plus Kc. K is usually a positive number if what we're talking about is non-ideality. And S will therefore decrease because this is a positive value. This will be bigger than 1, and so S will decrease. Okay. So S will have a concentration dependence. And so there will be this parameter K, which I would call hydrodynamic non-ideality, concentration dependence of S, that reflects something about its volume and charge in generality. It will be bigger depending upon how extended the molecule is. It will be bigger depending upon how asymmetric the molecule is. It might be bigger in a charge dependent way, depending upon what its charge is, because it's repulsive for other molecules of the same type. So this next slide shows some examples of plotting sedimentation coefficient versus concentration. And notice concentration here is expressed as mgs per mil, and consequently K, because it's a product, will be milliliters per milligram. So it shows 3 kinds of examples. So first, it's showing you sedimentation velocity for bovine serum albumin. This is a classic protein that people have been studying for 70 years. You can see it has a slightly negative slope. It's linear and slightly negative. This equation, if you look at it, really extrapolates to sort of a linear. Notice there's other terms out here, so it's possible at higher concentrations, you have higher ordered terms. In practice, you have to go to pretty high concentration to see that. With serum albumin, you probably have to go to 50 mg per mil to see that curvature. Another protein or another molecule on here is 5S RNA. And you can see it is steeply negative. It's nonlinear. So it has really, really high concentration dependence. It's highly non-ideal. And the reason for that is that it's sort of extended, but mostly because it's highly charged. Okay, so depending upon the salt concentration, this is really repulsion between the 5S RNA molecules. You'll notice that all of this non-ideality is happening below 1 mg per mil. The serum albumin, you don't really start to see a significant change until you get above 2 mg per mil or so. And so depending upon the units of stuff, for a typical globular protein, this isn't often very big. But the 5S RNA and RNA DNA in general is highly non-ideal. Okay. It absorbs very strongly because it's a nucleic acid, and so it makes it ideal for studying low concentrations. In the equation up above, there's this S naught, which is the extrapolated to zero concentration. There's an F naught extrapolated to zero concentration. Okay, and so generally speaking, what we want is the extrapolated value. And so we like to work at low concentration to get the extrapolated value for concentration independent, S naught, D naught, F naught, etc. The slope of the line gives you additional information potentially. Okay. A third example is glyceraldehyde phosphate dehydrogenase, which you can see they don't actually complete the extrapolation. They would have to have data points in this lower range. But what you can see is an increase in sedimentation coefficient. So what that suggests is that glyceraldehyde phosphate dehydrogenase is assembling into something. It's making an oligomer of some kind, some reversible associating substance, okay? And so lots of proteins do this, and you do sedimentation experiments in this way to get the concentration dependence. So in principle, as we'll see next week, you can use sedimentation data to solve assembly problems, okay? What is the stoichiometry of the assembly? What is the equilibrium constant for the assembly? What are the shapes of the oligomers? Those kinds of things. You would also like to be able to extrapolate back to zero to get information about the concentration independent term. That depends a lot on the shape of the curve and how hard it is to extrapolate. And the really hard part very often is the endpoint here. Where's it going? Is this really plateauing? Is that giving me the endpoint of the reaction? And that's really sort of one of the things that we need to and want to know. So velocity data, moving boundary data, tells you something about the macromolecule. Is it a monomer? Is it a non-ideal monomer? Or is it a polymer? And notice, both serum albumin and 5S RNA have a negative component. The fact that this is going up doesn't mean that there isn't non-ideality. It's just that you can't see it. You have to kind of tease that out from the data on top of the association. The association is dominant here, but there still is probably non-ideality that's measurable, and that contributes to the overall results. Okay, so the classic experiment about non-ideality was reported many, many years ago. There were a series of papers in the 40s and the early 50s on what became known as the Johnston-Ogston effect. And essentially, what you have is a concentration dependence of one molecule on another molecule. So here's a demonstration of concentration versus position. This is showing you the direct signal. So you can think of this as absorbance or fringes that give you some information about the concentration. You would have to translate this into the right units, and the figure has already done this for you. All right. And so there's a trailing boundary and a leading boundary. And we kind of sort of take a derivative of this, or we could do this with Schlieren optics. And you can see that there are two peaks that are sort of baseline resolved. This is a situation where there is no non-ideality, where there is so-called no Johnson-Oxen effect. In the second example, there's a Johnson-Oxen effect, and you'll notice that the area under the curve, the concentration of the slow boundary, is higher than in the figure on the left. And there seems to be more material on the left than there is on the right here. Okay, and so the reason for this is the material on the left sees the material on the right. Notice this is the sum of the concentrations. And so this fast boundary is actually sedimenting away from the slow boundary. If you really plotted just the concentration of the fast boundary, it would be down here someplace and it would sort of go down to 0 in here. But the slow boundary bumps up against the fast boundary, and there's concentration dependence of the kind that I just showed you in the S value, but it's due to the fast boundary. And so it causes it to slow down. It slows down in exactly the same form as what I have up above, this equation, except this equation has a term in it that is due to the effect of species 2 on species 1, and it depends on the concentration of species 2. This is the concentration of itself on itself. Okay, so this is the non-ideality of 1 on 1, and this is the concentration dependence of 2 on 1. And so there's a cross-term non-ideality caused by the second term, by the presence of the second species. Okay, and so this is the early indication that there's some kind of weird non-ideality going on. There was a lot of effort that sort of went into this. A lot of people published papers on this. And all it really means is that molecules in solution with other molecules can impact non-ideal phenomena on all the other molecules. When we talk about V bar, for example, it's the concentration, the volume change due to the adding of a molecule, holding all other molecules constant. Okay, but those molecules can interact with one another, and that interaction may impact things like sedimentation or diffusion coefficients. Okay. The first paper I ever published back in the 70s was actually a numerical study of the Johnson-Oxen effect. So we were looking at sort of modern ways of analyzing data and looking at 2 species Johnson-Oxen effects and trying to learn something about the system. I'm actually still working on this in a completely different setting. So this is a summary of a series of systems, including bushy stunt virus and hemocyanin and all these other proteins, showing you sedimentation coefficients. Notice they're extrapolated to 0 concentration, and they're corrected to 20 degrees in water. So the viscosity of water and the density of water, etc. The diffusion coefficient, the V bar. Okay, notice the bushy stunt virus, they're claiming it's about a 0.74. I think when we were calculating this, it came out to something else, but we were making some assumptions about the V bar of the protein. We didn't actually calculate it. And then this is showing molecular weight, and it's getting the molecular weight from what it says is S on D. The sedimentation coefficient divided by the diffusion coefficient gives you a D value. Okay, and so these are data that fairly old data, I guess, by older methods using Schlieren optics probably on all of these materials. The units here reflect something about how fast molecules actually sediment. This is corrected for 10 to the 13th because the proper units end up being S 1.14 times 10 to the minus 13 is the actual units that you get. Okay. The diffusion coefficient is expressed here, and this actually has the bigger units that we say. What we normally write is 1.14 S value, reflecting these units and reflecting the typical units that we like to use for sedimentation coefficient. The S stands for Sedberg. I'll say more about that in a second. The D value is multiplied by 10 to the 7th. And so that's because this actually has units of 10 to the minus 7th. It's usually in what's called FICK, F-I-C-K. So it's units of F after Fick's first and second law. Okay, and so that's usually the way we talk about these data. All right, and so these are typical data, and there's a relationship between the molecular weight and the S value. Notice that the S value is going up with molecular weight. The D value is going down with molecular weight. There's no obvious connection here to V bar because V bar depends upon amino acid composition or amino acid and RNA composition. Okay, but this is sort of raw data. Here's S, here's D, here's molecular weight. If we use Stokes-Einstein sorts of relationships and we write down Stokes' law, and then we use the A0 value here as though it were the total volume of the molecule. So assuming it's a sphere, because Stokes' law is for spheres, 4/3 pi r cubed or A0 cubed would be the volume of the molecule. All right. The volume of a molecule can be also written down as the molecular weight of the molecule times the V bar of the molecule, how many milliliters per gram. So here's grams times milliliters per gram, divided by Avogadro's number. So what that gives you, if you think about that calculation, is the volume of one molecule. Okay, the volume of one molecule. So we can solve here for A0. If we take 4/3 pi to the other side, so we end up with 3/4 pi, 3 in the numerator, 4 pi in the denominator. And then we take the cube root, that'll give us A0. And then we plug that into Stokes' law. And so we get this equation, 6 pi eta, and then the cube root of 3m V bar over 4 pi Avogadro's number. Then we're going to plug this into the equation for the sedimentation coefficient. Okay, so the sedimentation coefficient that we've used is m times the buoyancy term divided by nf, or f0 if it's a sphere. And now we've solved for f0, assuming that it's a sphere and that we can calculate the volume of a total of a single molecule, corrected for Avogadro's number. This is the mass of a mole of molecules. And so this is the equation that we come up with. And then if we move some terms around and we take S V to the minus 1/3 divided by the buoyancy term. So these are all basically experimental values. If you go back over here, you can see where this stuff is coming from. We're moving stuff from the right over to the left to come up with a slightly different equation. So the buoyancy term, and then an S naught term, and then it's corrected by the V bar raised to the 1/3 power, the minus 1/3 power. It was in the denominator over here. And we're going to plot that versus the molecular weight for molecules where we have experimental data. So like in this table, we're going to steal some data. We've got molecular weight, we've got S, we've got a V bar, and we're going to plot the data. So this equation suggests that S goes as m to the 2/3 power. Okay, and so by doing this as a log scale, because notice this is units of log, that 2/3 in front of the log will turn this basically into a straight line. And the line in the solid line is, as it says here, for an unhydrated sphere. The other data points are experimental. So notice the experimental data does not fall on the line because the line corresponds to unhydrated. What's missing here is a correction for water. All macromolecules have bound water to them. And so that's dragged along and it increases the volume. So there's so much water bound per gram of protein. So it's usually expressed as some kind of delta term, and it usually is grams of water per gram of protein. And this delta term is usually a correction. Okay, it's not a linear correction, but there's some kind of a correction for hydration. So all of these molecules fall on a line that's offset by some amount. Okay, the offset, I don't think they talk about the offset. The offset essentially talks about the amount of water that's bound, and they fall on a line that's basically off the line by a little bit. Okay, so what that means is that all of these molecules are globular and they have approximately a spherical shape corrected for water. It's not entirely true. You can see some molecules fall off the line a little bit more than others. There are exceptions, fibrinogen and myosin. Fibrinogen and myosin are rod-like. So not only do they have a correction for water, there's another correction that has to go in here for shape. So that's why when you collect sedimentation coefficient data, you get information that in principle gives you molecular weight. There's information about hydration that we often have to assume. We don't necessarily measure that. And then there's information about the shape. I said, I'm not sure what I just said there. I should have said hydration if I didn't. And then there's some shape term that you can potentially get that shows up in the F over F naught term. Okay, so rods fall off the line because they're dominated by their shape contribution. So in principle, this is showing you that there's a relationship across many, many proteins. Globular proteins will behave in a certain way, and you can use these data to infer hydration, infer shape, and measure molecular weight. Okay, and so you should be thinking about this as sort of a snapshot into the world of how do you interpret sedimentation coefficients. Again, notice S goes as 2/3 the molecular weight. Okay, so I showed you data a few moments ago about glyceraldehyde phosphate dehydrogenase, and it assembled into something. Okay, and so the question is, what does it assemble into? So on this slightly out of focus figure is some of the stuff that I just said to you. The F over F naught value is corrected for hydration and shape through terms like this. Okay, and what does it say here in this text? It doesn't say what I think it should say. So we assume the total volume of the hydrated molecule is simply the sum of the anhydrous volume and the volume of hydrated water. Okay, so if there's a water sum that goes into this, then the frictional coefficient will reflect a shape term, but it will also affect a certain amount of water. And here is the delta term that I just told you about. And you can see it's introduced as 1 plus delta to the 1/3. So this is sort of correcting for the volume of the water. Now, let's imagine that this molecule then made an oligomer, something that's bigger, an N. It could be a dimer, it could be a tetramer, it could be something big. So what's the frictional coefficient of the N-mer? Okay, that's that extrapolated data that I was showing you in the previous figure. That extrapolated curve goes to some value S, and we want to interpret that in terms of an oligomer value. Okay, and so we basically want to have some way of correcting for the oligomer size. And so a theory was developed in the 50s of a way of sort of predicting what that F value and then that S value would be. And it basically involved correcting the data for the distance between subunits, I and J, summing over all subunits, and then correcting for what the frictional coefficient of the monomer is, and trying to come up with some guesstimate. Okay, so that's not a great explanation of what's going on. This is sort of the beginnings of a technique that was developed. All right, and so if you have a monomer obeying this equation, okay, what is known as the Sedberg Equation, the molecular weight over an F value with a buoyancy term, that equation allows us to derive, a predicted S value for an oligomer based upon these sorts of calculations. You sort of have to know something about the shape of the oligomer. You sort of have to know, for example, is the oligomer, oh, I'm grabbing the wrong pen. You have to know, is the oligomer a tetramer? And what are the distances between these? Or is the oligomer a linear molecule? And what are the distance between these subunits, etc.? Is the oligomer some kind of a trimer, etc.? Okay, so these distances depend upon knowing this. And often you sort of assume that when you're analyzing data in the days before crystallography. If you didn't have a crystal, you would be able to use this to sort of guess. Okay, and so there's ways of sort of assuming something about the size of the S value and information about what the oligomer might be. Okay, and so here's a table that sort of goes along with what I just said. If we had experimental data, monomer, tetramer, and we knew something about the S value, so the ratio of S4 to S1, and that's what these are expressed as, ratios back here, then I could predict if it was a linear or a square planar or a tetrahedral structure, depending on how much bigger the tetramer S value is than the monomer. Okay, and so it was a way of sort of analyzing the data and gaining information about quaternary structure out of the S value. Okay. So let me say one more thing about this to sort of put this in a different language. If S goes as m to the 2/3 power, and this is actually an approximation that assumes a sphere going to a sphere, the monomer of S1 is a sphere, and the monomer of the tetramer S4 is also a sphere. This would go as m to the 2/3 power. This is basically saying not if it's linear, not if it's square planar, not if it's tetrahedral. So if this was 4 raised to the 2/3 power, it would be 4 times bigger. It would actually be 4m raised to the 2/3 power. So it would be 4 to the 2/3 power, m to the 2/3 power. Okay, and so this would tell you an equivalent size if it were a sphere, which it's not. And I'm opening up my phone to calculate something. So I'm going to my calculator here. So it should be about 2.5. If it's a sphere, 2.52 is the number that I'm getting for this right here. Where's my pen? 2.52. Notice tetrahedral is 2.5. So tetrahedral is approximately a sphere. I can't draw it real easily. It's a flat bottom with one in the middle and the top, creating a pyramid, if you will. So that's close to what it would be. If the S value for the tetramer was less than that, then it would be telling you something about the shape of the tetramer, okay? So here's an example of that for so this is hemocyanin. I'm looking at the figure legend, and it's simply showing you the relationship between, I'm not sure if this is monomers. I think this is an oligomer interacting with an oligomer, and the 17S going on to make a 39S. So this is saying this is not reversible. So this oligomer is stable. This oligomer is stable. They're probably made from one another, but kinetically they run separately. Okay, so this is transitioning to something else. But you can imagine if these oligomers were interacting with one another, then the increase in S would tell you something about what it is. Okay, that's a little more than if it's non-associating. So it's 39 over 17 extrapolated. 39 divided by 17 is 2.29, 2.3. Okay. And so depending on the shape, this could be something like a dimer of this. If they're not reversibly interacting, but they're assemblies that are stable, then this could be a dimer. It's in the right ballpark. 2.29. I'm sorry, that's much bigger. 2.29. Go back. 2.29 puts you right here. So this actually is probably close to a tetramer. It's 4 times bigger. If it was a dimer, it would be 2 to the 2/3 power. That's 1.58, 1.59. So running this much faster, this is actually a tetramer. So this is probably a tetramer, and I just showed you how you would reason this out. Okay. The other thing I should point out here is this is S on D. Notice the S equation is up here in the numerator, and the D equation is down here in the denominator. They both are divided by NF, so they cancel. And what you get is M1 minus V bar rho over RT. So that table up above said S on D. So what it's doing is it's using the S divided by D result to calculate a molecular weight. You simply need the buoyancy term. The V bar, the rho, R and T are constants. You know what the temperature of the experiment is. Okay, so this is how you would solve for a molecular weight from a sedimentation experiment. You would actually measure the sedimentation coefficient. You would determine the diffusion coefficient. Notice that this one is sharper than this one. So this is significantly bigger than this one because it's spreading more. Okay, and then you can get a molecular weight out of this. All right, so the fact that these are not associating and not what we call an interacting system means the shape of this corresponds to a single species with a known molecular weight, a buoyancy term, etc. Okay. All right. Now, depending upon how I went about teaching this kind of stuff, what I would do is, and in different books, and I think this is something that I stole from a different edition of Van Hol because he didn't have it in this chapter. We would look at the chemical potential. So the way we solved the osmotic pressure problem, remember, we did the chemical potential, but because the protein was in there, we had to add a pressure term, and we needed another term, and we eventually solved for the osmotic pressure, which gave us some information potentially about molecular weight. In a sedimentation experiment, the total potential corresponds to the chemical potential of the solvent, the solute, excuse me, plus a term that corresponds to the fact that it's a spinning rotor and there's a force on the macromolecule. Okay. So the dependence of the total potential on the position in the cell as it's spinning would be the partial derivative of the chemical potential with respect to R. And so the first term would just be this derivative, partial of mu with respect to R. The second term would involve the derivative of R squared, so the 2 comes down, and we would just get minus M omega squared R. All right. This would be a way of describing what is known as sedimentation equilibrium if we set this equal to 0. If you spin the sample long enough, generally at a lower speed than you would do sedimentation velocity, this would come to equilibrium, reflecting the influence of the change in chemical potential that's due to the concentration dependence, and then the change in the chemical potential due to the force from the centrifuge field. And so we could plug this into the flow equation that we used up above in terms of this linear connector and this potential, which we then just plug into this equation. Okay. And then we write the whole thing out in terms of all of the dependencies. And notice the way this is done as you expand it. The temperature dependence would be d mu dt, dt dr, and then d mu dp, dp dr, and then d mu dc, dc dr. Okay, so the temperature dependence would be 0 because all across the cell, there is no change in temperature with radius. If there is, there might be convection, so that could be a problem. The pressure dependence of this reflects something about the pressure dependence due to the increased distance away from the center of the rotation and the fact that there's more force on the solution. So you would have some kind of a pressure dependence that's shown down here below. The pressure dependence of the free energy is equal to the volume, it turns out. You have to go back to some of the stuff we derived in the third or fourth lecture. And then expressed as a chemical potential, we divide by N. So this now becomes a partial molar volume instead of a volume. Okay, and so this term can be interpreted. And then the last term is RT over C, dCdr, and this has something to do with the concentration dependence of chemical potential. Remember, chemical potential is mu is equal to mu 0 plus RT ln C. I wrote that down, I think, somewhere in a previous slide. So go look for it. And then if I take the potential of this with respect to C, I get 1 over C dCdr. So that's where this term comes from, taking the derivative of this with respect to C. Okay, and that's shown at the top of this slide here. That's where that term comes from. This is walking you through what the pressure dependence is in R. And so you end up with this equation involving the flow, omega squared R times the molecular weight and a buoyancy term. Okay, and then here's the other term involving the concentration dependence. The connector from up above is C over NF. And so we can plug that into this total equation, and we see that the flow is equal to this molecular weight term times this omega squared RC, and then this RTNFC. The C, notice here, cancels the C here. And so this is the equation that we get for the flow. We did a similar thing for the diffusion coefficient. And now we're going to set that whole thing sort of equal to 0. All right, so what this is doing is it's actually deriving a different equation. So we now have the flow in terms of a sedimentation coefficient. Notice this is the equation of a sedimentation coefficient, and a diffusion coefficient. Notice this is the Einstein's equation. Okay, so the flow can now be expressed as S omega squared RC, where this is the value of S and this is the value of D, minus D, dCdr. Okay, and then we can plug the flow into what we called previously the continuity equation. dCdt at constant R is equal to minus 1 over R, drj, the flow, dr constant T. It's 1 over R is because it's a sector-shaped cell. So we're correcting for the change in volume at higher R. Okay, so plugging J into this equation, this is what we get. Okay, dCdt at constant R minus 1 over R, and then this double derivative where there's a partial with respect to R, this extra R term multiplies through, and so we end up with an R squared and we end up with a dr. This equation is known as the Lamb equation. This describes simultaneous sedimentation and diffusion in a field during a rotating field in the centrifuge. What's the change in concentration with time in terms of the change in concentration with position? There's a sedimentation that occurs, and there's boundary spreading that occurs once you have a gradient. So this is the way that we would generally solve what's happening in that boundary. And I think the next figure is going to show us, well, these figures are going to show us something about that. Now, it turns out that this equation is hard to solve. And that paper that I showed you from 1976 was one of the early examples of how we solved the Lamb equation and then applied it to a two-component system. There was a C1 and a C2, and then there was an introduction to concentration dependence in the S and the D as well. It was sort of the beginning of what I would call modern sedimentation analysis. Okay, so this Lamb equation is what we actually use today to solve sedimentation velocity problems. This is the equation that we write out, and this is the equation that we use, okay? There are lots of different ways of analyzing data. One of them is the so-called Van Holt-Weisschit method, which was a paper that Ken Van Holt published in the 70s. And it was a way of analyzing the shape of the boundary in terms of a sedimentation coefficient. And you basically looked at little regions of the boundary, extrapolating the data back to 0 time, and you could get some information, maybe it was infinite time, extrapolating the data to infinite time, and you could get information about the S value. In this particular case, the S value gave you data suggesting a single species, okay? And so that's what this technique was used for. Another way of analyzing this is to fit the data as a series of solutions to the Lamb equation. It's known as a C of S. It comes from a program called SedFit. And it allows you to look at a boundary and ask, are there multiple boundaries within the boundary? Okay, and so you can see in this particular example, this is serum albumin with a certain amount of contaminating aggregated dimer and trimer. Okay, you'll notice that this has been converted to molecular weight so that M to the 2/3 analysis that I told you about, this is actually analyzed as S, and then it's converted to molecular weight. And so the molecular weight of serum albumin is around 66,000. So you can see there's 85% monomer, there's 12% dimer, there's 2% trimer, and there's a little bit of breakdown product. This is some kind of, I'm not sure what it is. It looks like it's running at about 25,000. So it's some kind of cleavage product. You would think if there was this, there would be a other part of a chain. It's not exactly half of this, but it's allowing you to see that it's a mixture of stuff. This is solving this analysis using this kind of equation. I'm not going to go through all of the technique that is used in this method here. Okay, so this is sort of the way that we analyze data this way. Okay, and again, this is showing another summary of different kinds of molecules, including a polymer, their S value, their D value, their molecular weights. It's showing you some temperature dependence that might be going on. Okay, but again, it's giving you information about the relationship between S and M, D and M, etc. Notice something. I meant to say this a little bit earlier. If M, excuse me, up here, where's the pen? If S goes as the 2/3 power, but S on D gives you M, then this must be M to the 1/3 power. Okay, because the combination of this gives you M to the 1st power. Okay, this gets brought up into the top and we can actually add these together. So S goes as the 2/3 power for a sphere. D goes as the 1/3 power for a sphere. That dependency varies with hydration, it varies with charge, it varies with shape. Okay, but generally speaking, those are simple starting points for interpreting S values and D values and their molecular weight dependencies. Okay, and again, this is S for units, Sedbergs. This is F for fix. These are the units of fic. Okay, so that's the way that we would normally talk about this.
Dr. Correa,
Yes.
okay, in that slide that you just showed for the M, for the denominator, it will be M to the power -1/3, isn't it? If we want to get M, because when it goes up, then we kind of add to M to the power 2/3.
You're right. So,
Plus.
so the way I'm writing this down, I'm making a mistake, aren't I? D goes as M to the -1/3 power.
1/3, yeah.
Yeah, and that way, when it comes up,
Then it will be the
and then it
addition.
will
Okay.
be S on D will then be M. Thank you.
Yeah.
Okay. All right. I'm going to say more about that analysis if and when we have time. I'm not sure we're going to have time. I think we'll try to talk about it a little bit at the very end and then some next week. Now, if the flow written here gets to a point where it stops sedimenting, so you run it at a slower speed and at time 1, there's some kind of a sedimentation that happens, but eventually it stops sedimenting and it sets up a constant boundary. This is that chemical potential that reflects a concentration dependence of the solute and the chemical potential due to the total potential due to the field. This shape is what we would get as we go to sedimentation equilibrium. This is where essentially the flow goes to 0. Okay, so if the flow goes to 0, okay, so this slide is sort of doing what I was starting to do in my head back here. Okay, let's use this as a better outline so that I don't make a mistake. So here's the flow due to velocity. And here's the flow due to diffusion. And we're going to set these equal to 0. So that must be, I must have grabbed a slide where I show the diffusion flow, and then I show the velocity, the sedimentation flow, and then we sum them together as a total flow and set it equal to 0. And then we rearrange. Okay. When we rearrange, we plug D in as RT over NF, and then bring this to the left. So it's RT over NF dC dr. And then it's equal to M1 minus V bar rho omega squared RC over NF. The NFs will cancel. Can you see that? And so we'll have dC dr. This C can come to the other side. So the NFs will cancel. This C will come to the left. And so we'll have dC dr 1 over C. This RT will come to the other side. dC over C is dlnC, because dlnC is 1 over C dC. So this becomes this. Notice the dr changes and it becomes dr squared. Okay, so dr squared is 2r dr. There's an R right here. So this has to come to the other side as well. Okay, so when you bring this r to the other side, you have r dr. Okay, 2 dr squared is 2r dr. Okay, so we're going to convert this to dr squared. There's an extra 2 here someplace. Okay, and so that extra 2 has to be accounted for, and that's what happens. We sort of multiplied and divided by 2, and we end up with an extra 2 to get rid of the 2 that's missing. Okay, and so the equation ends up being d ln c dr squared is equal to m1 minus v bar rho omega squared. Remember, omega is 2 pi rpm over 60, angular velocity, divided by 2r squared. Now we're going to integrate that. And when you integrate this, we're going to bring the dr over here to this side, and we're going to integrate the dlnc. When you integrate dlnc, you simply get the natural logarithm back, and you have limits of r0, the meniscus, to some position r, and you go from r0, you go from some concentration at r0 to some concentration at r. And on this side, you go from r0, the meniscus, to r. Okay, and so the integral of dr, okay, so I just used a lot of language here to explain this, but this I think will be clearer. This will be the integral of dr squared. Okay, going from r0 to r. Okay, and so you basically get r squared evaluated at r0 and r. So you get r squared minus r0 squared. And that's what gets multiplied by this other term in the front. And then on the other side, we have the integral of d ln c from c0 to c. Okay, and so you get ln c evaluated from c0 to c, and that's why you get ln c over c0, because it's a minus c0. And so you get this on the left, you get this term corrected for that on the right. That's the equation for equilibrium. That's the equation for equilibrium. And then we usually raise it to the power of E. When you take E up, when you take E to the natural logarithm of C over C0, that ends up being C over C0. This is the opposite function, right? And then you take E raised to the power of everything on the right here. And so that's where you get m1 minus v bar rho omega squared 2rt times this term. All right, so I usually write this as e to the sigma psi, where sigma equals m 1 minus v bar rho omega squared rt, and psi is r squared minus r0 squared over 2. You can see the r squared over r0 squared over 2 in the term. This is known as the reduced molecular weight. And what you can see is it involves molecular weight and speed. Okay, so the curvature that you see, the dependence of absorbance or signal on position in the cell, how curved this is, how shallow it is, how sharp it is a measure of sigma. We usually want sigma to be somewhere between 2 and 3, 2 and 4, and you get different sort of shallow curves depending upon what the value of sigma is. It can be shallow, it can be steep, and that's generally reflected in this sigma value. We use this to choose speeds. Okay, and so I actually calculate sigma depending on molecular weight to get a value. Usually I'm looking usually for a value of 3. I'm usually going from 2 to 4. I choose speeds to give me a certain amount of curvature. If I plot this as natural logarithm of C versus R squared, that's the equation for a straight line. The slope of the straight line gives me the molecular weight. So if I plot this as the logarithm of the absorbance or the signal, corrected for some baseline offset, there's usually some noise in the baseline, versus this R squared term, that's the equation for a straight line, and the slope of this should be proportional. Sorry, I'm trying to make this. The slope of this should be proportional to sigma, should be proportional to molecular weight, corrected for all of these other terms. Okay, so the old way of analyzing data was to plot ln versus R squared, and the slope of the line gives you the molecular weight. What we really do is we fit data that like this to this equation to get the molecular weight. So we actually fit for this sort of analysis to get a molecular weight of the equilibrium constant. This is simply showing some examples from the literature of sedimentation equilibrium experiments where we know what the molecular weight is from a chemical formula. We know what the molecular weight of sucrose is, and this is what somebody measured. We know what the molecular weight of ribonuclease is. This is what somebody measured, okay? For example, okay? And so you can do a pretty good job of getting molecular weights by this type of approach. Notice when you're doing this, there's no flow and there's no shape because the NF terms dropped out. So if you have a tetramer that's linear or planar or tetrahedral, this experiment won't necessarily notice that. It will notice it in terms of some non-ideality effect and some volume effect that might show up in a virial coefficient, but it won't notice it in terms of just a straight molecular weight because this doesn't have a shape term in it. Okay, as a first order effect, it doesn't have a shape term in it. Okay, if you have a mixture of terms, then mixture of materials, then what you will see is a total concentration summed up. This is what the signal would look like, but there would be a contribution from the small component, a contribution from a big component. And when you plotted LNC versus R, you wouldn't have a straight line. It's not entirely obvious to me that this isn't a straight line, but a straight line might look something like that. Okay, so the curvature tells you there's a mixture in here. There's something going on, and you potentially can sort that out from an equilibrium experiment as well. In a velocity experiment, it would be easier because you would see two boundaries potentially. Okay. If it was non-ideal, let's say it was a single component with non-ideal, then this would curve down. So curving up would tell you there's a second component or there's association. If it's curving down, then there's non-ideality. Okay, this would be showing up in a virial coefficient. Okay, the same kind of term that we talked about in the Donen effect. All right. And so when you collect data like this, you're looking for purity, you're looking for lack of heterogeneity, you're potentially looking for association or non-ideality. And again, everything that associates into an oligomer will also be non-ideal. You potentially have to sort this out, and you essentially get both terms at the same time. You have to be able to fit for both of those terms. Okay, so what he then does in the book is he shows you the equation that we just wrote, dln CDR squared in terms of a molecular weight and a speed dependence. And then he says, okay, if it is a mixture of things, then what you'll get is a weight average molecular weight. And remember all the way back to the table that we constructed from the osmotic pressure section where we looked at number, weight, and Z average molecular weights. And so you'd get average values, and you can interpret data in terms of weight average molecular weights. And my computer didn't want to move forward there. I think that's a blank slide. All right, so let's take a few minutes break. And we'll discuss a couple of different techniques, and then we'll very quickly discuss a couple of historical articles. So let's take a couple minutes break. Our usual 5 or so, 11:10, 11:11, we'll come back. Okay, let's get going. Or is everybody back? So in that technique that we just went over velocity, we went over equilibrium. What I want you to appreciate is that you start out with a constant concentration. So everything is at the same concentration. And then when you start spinning, you get a boundary that sediments away from the meniscus, and you get material that pellets. And then over time, it establishes a constant gradient. At lower speeds, sedimentation equilibrium speeds, sigma's around 3, you'd get some kind of a gradient. If you spin really fast, everything would be as a pellet in the bottom. It would be 0 with a pellet. Okay, and so this is the way we typically do sedimentation velocity, sedimentation equilibrium. The main difference is usually how much volume we put in the cell. There is another way of doing these runs, and it has to do with what they're calling zonal sedimentation, where you have some kind of a material with a layer of macromolecule on the top. Okay, and this material down in the bottom is usually density stabilized. So this is usually sucrose. It might be something else. It might be cesium chloride. Okay, it's usually something that will establish a density gradient so that the stuff that sediments will slow down. The density gradient that's in here is really to minimize the amount of spreading that goes on in these two zones. So in this particular case, it's simulating two different components that sediment with different S values, okay? And what it means is you can capture them, all right? You can capture them and isolate them in principle. So by observing them, you say, ah, look, we have two components. We have a slow and a fast sedimenting material, and you can sort of get information about it. These cells are usually plastic. You can usually puncture them, and you can usually draw off material and then capture the various peaks. You can, as you puncture them and you draw off material, you can flow it through a spectrophotometer, and you can get a signal, how much material. You could run it through a counting machine if they were radioactively labeled, etc. Okay, and so this is a way of fractionating and isolating materials. A lot of people used to use this technique. I don't know that many people who still do it, but I'm sure there are some people who still do this. Okay, and so this is basically showing some cartoons of what this is about. So here's an example of making a gradient. So they're actually preforming the gradient. You actually have a gradient maker. Maybe you've seen these devices, high concentration, low concentration. And as you pour it into the cell, it starts out high in sucrose. And then as it gradually fills the top, it becomes low. And so this gradient basically is something that you can use for fractionating. And then the cells are usually held in a swinging bucket rotor. And it usually runs this way where it swings out under the force so that the samples aren't banging into the wall as it sediments. If you were doing a fixed angle, you were doing some kind of a fixed angle rotor that might look like this. The tubes would look like this. The force would be that way. And so things would actually run down the wall. This way, they just run smoothly through the gradient. And then you get boundaries that align with the S values, how far they've sedimented. You have to know how long the sediment, and you can capture things. This is the technique, for example, that was used to isolate polysomes early in the 60s. This is the way that people said, oh, look, we have molecules with multiple ribosomes on them. What is that? Okay, so this was the technique that was used for that type of an analysis. And then this is showing what I just described to you, a way of fractionating the gradient, the plastic tube allowed to drip, and then they can be assayed. So you're collecting different volumes. These volumes look pretty big. You could do this as only a few drops or more. Okay, and then this is an example of the way you might do analysis of different materials that had different S values and fractionate across the bottom. This was described in the very early 60s, and I'm not going to go into any of that, by Martin and Ames. And so there was a lot of this done in the 60s and 70s on various results. All right. I can tell you stories. I'm not going to, from when I was a graduate student. Okay. So that's one of the things that you could be doing. We do, in fact, have a swinging bucket rotor, and this technology still exists. You can still buy swinging bucket rotors from Beckman. So there may be reasons for doing this. Okay. The other technique that could be done is to not preform a gradient, but to allow a gradient to form while you're spinning. And if you allow the gradient to form while you're spinning, shown here, you would get a banding pattern where the material would band at its buoyant density. Now, sucrose will not work for proteins or for DNA generally, okay? But if you use something like cesium chloride, you can establish a density gradient being established in the back due to the cesium gradient, and then the protein would find its natural place where it's buoyant. It doesn't sediment or float, it stops sedimenting. And if you go through the mathematics of that, you discover that the width of the boundary is something that gives you information about the molecular weight. Okay, because there's a square root, inverse square root of the molecular weight. There's something in it about the density gradient. The buoyancy term is the term that determines the V bar is the term that determines where it's going to migrate. Okay, and so this is density gradient sedimentation. Sedimentation equilibrium and a density gradient gives you the ability to fractionate material by its buoyant density. All right. Satellite DNA was discovered this way. Look at this funny thing that's sedimenting at a different density. What is that? Okay, it's DNA that has a dramatically different V bar, a dramatically different sequence. Okay, and so that's that technique. I'm going to ask you when we talk about this a little bit, if you know about this method. In the late 50s, mid-late 50s, Messelson and Stahl used this to study replication of DNA. You know that in the mid-50s, Watson and Crick published a famous paper telling us what the DNA structure is: double-stranded, anti-parallel helix. That suggested something about mechanisms of replication. Certainly suggested something about genetic duplication. But it didn't actually tell us how DNA replicated. There was a whole lot that was not known in the mid-50s. Okay, and so Messelson and Stahl decided to use a density gradient technique, and they grew bacteria in the presence of N15 and N14 nitrogen so that they could maybe do a sequence of timing of replication and watch the DNA replicate, and then band it out in a sedimentation equilibrium experiment in a density gradient technique, and watch the initial DNA formed incorporate a different nitrogen isotope and learn something about the mechanism of DNA replication. And so that figure that's shown there is something that's in the book, and I have stolen it, but they stole it from a paper. Okay. And so the data actually looks like this. You can look at this. See these doublets here? doublets here. This is the figure out of the paper that Messelson and Stahl published. So the idea was is that if you grew up DNA in the presence of one isotope and then let it go for one generation and then resuspend it in the presence of a different isotope, okay, so it would be N14 or N15 containing materials so that you would make the DNA out of those bases with the different isotopes and then fractionate it in a sucrose gradient at some speed for some time. You would see two different bands depending upon the N14, N15 composition. So the idea was is that generation one would grow up with certain isotope and it would give you a band that migrated to a second band as it incorporated a different isotope. Okay, so you grew it up in one isotope and then you changed isotopes and you allowed it to go through another generation. So the bacteria obviously had to be sequenced, had to be timed. And what you can see is that there's a gradual shift from a single band to a doublet that then becomes a single band. So at the end of the first cycle, everything converted from one density to another density. And you can see the transition going from here's the initial, and then something's being made, and then eventually you get something else. And then you do a second generation, and then suddenly what you see is a doublet. You see this band, but it migrates now with a second species, and you suddenly at the end of two generations have two species. And then if you keep going and going, you then begin to see that you have an increase in this leftmost band and then a residual band here on the right. So this is sort of what the raw data looks like. We're starting out with N15 labeled nitrogen, the DNA, and then you let it grow in N14, and then suddenly there's another band. And then if you keep letting it grow, there's a pair of bands, one of which grows and one of which basically stays the same. What does that mean? So it turns out that the original parent band in N15 gave a single band. But then when you let it grow in N14, what you got were a different band, but it seemed to be the old initial strand with the new strand, the old initial strand with a new strand. So you now had a species that was a mixture. The first generation after the labeling was a mixture of the old strand and a new strand, the old strand and a new strand. And so this was the original evidence for semi-conservative replication, that something machinery came along and duplicated each strand separately and made a mixture of strands. And then if you kept going, you would still see mixtures, but now these new strands would also be made with N14, and they would have a different density than the first generation. And that's why this would grow in amount, because the next generation, the third cycle and the fourth cycle would be all N14, and the original mixtures would still be there. And so you would always see them in the experiment. And so this is depicted as DNA, double strand, one strand is duplicated, the other strand is duplicated, and then it goes on, okay? So this was sort of this was the first evidence of what the duplication process was. Semi-conservative replication meant that each strand was going to be copied separately. And you could fractionate them in the centrifuge in a cesium gradient, and you could see that when you switched from N15 to N14, you suddenly got strands with different densities, but those different densities were different from the N15 strand. And then you got something that was completely different because now these were all N14. okay? And that's why when you mix different materials, you see 3 total bands. You see the N15 band, you see the N14 band, and you see a central band, which is the N14, N15 band. So they're mixing material from 0 and 1.9, and you can see there really are 3 different species. The original, the semi-conservative replication, and the complete N14 material that you get in subsequent generations, okay? So this was the evidence for semi-conservative replication of DNA. Now, there's a lot that we, of course, didn't know, and a lot had to be learned, but this was sort of the unknown in the DNA hypothesis that Watson and Crick proposed. You could have imagined something completely different than this. We can't any longer because of what we've all learned and what we know in textbooks, okay? You could have imagined that somehow this didn't have this intermediate step, okay? That there was no semi-conservative, that somehow each molecule was completely replicated and the original material somehow got lost. That doesn't really make sense to us anymore, okay? But that's one possibility. Are there any questions about this? Have you been taught about Messelson-Stahl?
Not me, Dr. Correa, and I will have questions ultimately, but I need to spend a little bit more time on it with myself, I think.
So one of the things that's happened over the years, I said this to you early on, everything in textbooks comes from something, comes from a paper, comes from a discovery. And this is one of the things that is no longer in textbooks and it's no longer taught. It's like, yeah, replication is semi-conservative. We don't really care. We're not going to teach people the method that allowed us to draw this conclusion. Okay, all of the other details are more important. How do you do the replication? How do you do the translating of the replication of the material with different kinds of fragments and how you replicate each strand separately at the same time, blah, blah, blah. All of those details are sort of in a textbook, but you don't necessarily discuss how it was done, okay? And I think it's important because this is a lecture on sedimentation, that you see that and that you know that it came from this. I have a slide back here. There's a textbook that, there's a book that appeared about 20 years ago. The Most Beautiful Experiment in Biology was the title of it, okay? And it basically described who Messelson and Stahl were. I think actually, I'm not sure that they're dead. I think they may still be alive. And how they met and how they arrived at designing the experiment and how they did the experiment, et cetera. Okay, so it's sort of a, you know, a 50s level picture into what the early days of molecular biology were like. So it's a nice book if you want to someday in your spare time discover it. Now, the final thing I'm going to talk about here real quickly is this review article that Ken wrote 20-something, 25 years ago. This is an unusual journal that's published. This issue was put together by Jeff Hansen. I actually have an article in this issue. And it's basically the initial history of ultracentrifugation, analytical ultracentrifugation. And it all basically, oh, I know what I want to do here. That's the last slide. Ha. I wanted to go to the paper. So it's basically giving you the history of where the method came from. And the method came from this guy called Sedberg. Remember, the units of S are Sedbergs. And he spent his entire life and career in Uppsala in Sweden. That's the big university town in Sweden. And he built the first analytical ultracentrifuge. There's a huge history on this. This book describes that history. I actually have that book in my office. And what he was studying at the time in the teens and 20s were colloids. And he was interested in this idea that proteins were actually colloids, similar to the kinds of colloids that he studied in his chemistry labs. And he was actually doing sort of diffusion experiments and trying to understand colloids. And the field at the time thought that proteins were colloids, just mixtures of components that were held together by some strange non-covalent forces. And this was at the beginning of us isolating proteins. And so they started sedimenting proteins, and they started sedimenting stuff like hemoglobin. Okay, and as they started doing these experiments, they discovered that they weren't colloids, they were clean macromolecules. Okay, and so they did hemoglobin. There were a lot of other techniques that they used, but they did experiments, and they basically did equilibrium experiments as well as velocity experiments. And what they discovered was that it was a single component with a well-determined, you can do the statistics on this, well-determined molecular weight. Molecular weight of hemoglobin is around 68,000. And because of osmotic pressure data, they knew that this agreed with the osmotic pressure data. And then they actually had some iron analysis data showing that there were 4 iron centers. And so they began to realize that, gee, maybe there are subunits here that are 1/4 the molecular weight. So they were able to do things on the subunits and get information about the composition of these macromolecules. Now, the thing is that you need to appreciate, at the time we thought proteins were colloids. And now here comes along these observations. No, a protein is actually a single composition. But wait a minute, this one is actually made up of subunits. So the whole idea of primary sequence and quaternary structure had to come out of this. What is this thing? Why is it a single component? Where does it come from? How are there 4 subunits in there? How do they assemble? Why are they stable? This is the beginnings of biochemistry. We would call this the beginnings of molecular biology, but this is basically the beginnings of biochemistry, that each protein is a unique thing. We have no idea where that sequence comes from. We have no idea how well determined it is, et cetera, but they were able to determine all of this. And so they did a whole series of experiments on mixtures and contaminants, and they would do velocity experiments where they would try to analyze the data that they would collect. I won't go into the optics. The optics were really sort of strange knife edge optics where the light would disappear where their high concentration was. And so you could watch the light reappear because there was too much protein in there. And so you could sort of expose a film and you could watch material sediment. And then they developed ways of analyzing those sedimenting boundaries. The Lamb equation was actually derived. You can see the name here in that period of time. You can see that Thesalius was around at that time, coming up with electrophoresis techniques for studying these same kinds of macromolecules. You can see that there were techniques. This was in 1929. There were techniques for looking at hemocyanins already. Sedberg was actually looking at hemocyanins, a different kind of oxygen binding material. And notice that there are multiple boundaries in here. So we're already beginning to learn something about the idea of subunits and different oligomers and maybe different assembly pathways. we didn't really have the language for this. I want you to appreciate that we didn't really understand this at all. This was pretty revolutionary. Sedberg also got the Nobel Prize for this work, although initially he was being given the Nobel Prize for colloids, but his Nobel Prize speech actually talks about all of his centrifuge work on proteins. Okay, so the idea of primary structures, quaternary structures, reversible assembly all came out of this work. Okay, and so everything that I sort of do and that we do today was already sort of known and beginning to be talked about. What was lacking was easy instrumentation. He had basically the only working centrifuge in the world. That's not entirely true. There was a similar instrument in Wisconsin where he went and spent a year on sabbatical and then went home and designed a bigger instrument. It wasn't until after the Second World War that the quote unquote Model E was invented by a bunch of investigators, primarily in the United States. And that's the instrument that I actually grew up with in the 70s. It was from a company called Spinco that became Beckman. And that was the original technique that most of us used at that time for doing analysis. And the text in here is describing all kinds of stuff that was done. They just show one example here. This is actually supercoiling, and this is showing you nicked DNA in black and supercoiled DNA in white. And what they're doing is they have some kind of supercoiled DNA SV40, which some of you may know, and they're adding a drug to it that unwinds it. So they're adding a ethidium usually, and it causes an unwinding. And as the DNA unwinds, it goes from something that's really tightly wrapped to something that's more extended. And so its sedimentation coefficient is fast because this is compact. It then slows down because this has a bigger frictional coefficient. But then if you keep adding ethidium, it starts to supercoil in the other direction. And I can't show you that here. And so it rewinds. So this must start out as negative supercoil, and then it becomes positive supercoil. The negative supercoiling is relaxed until the data coincides with the nicked DNA, and then it leads to positive supercoiling, and the shape changes. So the molecular weight never changes, the shape changes, the F over F naught changes. Okay, and so this method, the sedimentation method, was used for this type of analysis. There are lots of other stories that can be told about the entire history of the centrifuge. There are lots of us who have written lots of different methods on this. I just wanted you to know something about the original history, which primarily is Sedberg and the original idea of primary, secondary, tertiary, quaternary structure. This is really primary, quaternary, and reversible interactions. Okay, let me get rid of that. Yes, you can keep whatever I wrote on it. Okay. So we have a few minutes. What I want to impress upon you is that everything that I just said to you in the last 4 hours or so is sort of a traditional way of presenting sedimentation diffusion. And the other thing that has sort of happened in the field, you guys are way down here, is that people don't teach this in their graduate programs any longer, unless there's somebody in the department like me who does this method. This is a method that I use, okay? And so this is the traditional way, an introduction to diffusion, an introduction to sedimentation, an introduction to the classical methods that people use and the kinds of questions and problems that they solved. All right. The way I do sedimentation today, and it's explained a little bit in the Van Hold history article, that there's a new instrument out there that we all use, and that new instrument actually, there's a new generation of that instrument. We're all doing things very differently today because we no longer collect data on film, we collect data on computer files. And once you have computer files, you can collect a lot more data and you can do a lot more stuff. So the stuff that we do today, is this really where I am here? I guess this is where I am. Is analyzed with completely different kinds of technology. And that technology allows us to do sophisticated sort of analysis. And I'm going to, in 10 minutes or so, 15 minutes, show you what I mean. So this is a module that I can use for analyzing or simulating data. So I'm going to simulate data. I'm going to call this new, and I have no idea. I'm just going to call this, create a new directory, and I'm going to call this directory 2022. I want to create it, and then I'm going to say simulate tubulin SV. So I'm going to simulate a protein, tubulin, by sedimentation velocity. The model that I'm going to use is a monomer model, so I'm just going to call it A. I'm going to collect 500 data points. Oh, something changed here. Go back to simulate. The molecular weight of tubulin is approximately 100,000. The sedimentation coefficient is 5.8. The density increment is the buoyancy term. So I'm going to call that 1 minus V bar rho. I'm going to call that 0.3. So that's a V bar rho of about, and that's probably better at 0.27. The extinction coefficient is 1.44 per mg per mil. Okay, so if I have a 1 centimeter path, 1 mg per mil tubulin has an absorbance at 280 of 1.2, and the path length in the cell I use is 1.2 centimeters. So the product of those is 1.44. I'm going to simulate absorbance. I'm simply going to say sim tubulin at 1 mg per mil. I'm going to put the meniscus at 5.9 centimeters, 5.9 centimeters. I'm going to run it at 50,000. That's going to be okay. I'm going to put the base at 7.2 centimeters. I'm going to put a loading concentration of 10 e to the minus 6 moles per liter, which will correspond to 1 gram per liter or 1 mg per mil. So you notice I can change units here. Okay, so it knows concentration. All right, I'm doing velocity. It's telling me how many scans I need to collect. I'm going to make that a little bit bigger. It's telling me that I'm actually going to output a grid of 1,500 points. So I'm going to actually collect 1,500 points here. This is pure data, and this is basically going to simulate using the Lamb equation, using this information. I'm going to add noise to it. I'm going to add a certain amount of noise to the absorbance data. And then this is saying how long the experiment is going to last. It's going to be about a 6-hour experiment if I did it in real time. And now you're going to watch it happen. Boom, there it goes. So notice it pelleted because I chose a fast speed. I don't know if you saw it, but as it sedimented, the boundary spread and then it pelleted. Okay, so let's go back and let's do something that I forgot to do. Let's pull all of that back because it has a memory. And let's come over here and look in output file. And I want no, that's not it. I want to go here. Simulating. I want to output. Oh, it did. It output a data set. Okay, so that's what I needed. It output a data set ABR file. It didn't output the data set. I was going to have to go create something in a different folder. So it's created a single data set at one concentration. Okay, so let's cancel this and save it. Now let's come up here and process it. So let's go here and find the data. There's a data set out there. Notice there's 150 scans. So if I go here, it's actually processed, and notice it says simulated tubulin SV. Okay, so it wrote the data, but you can't see it because it scaled the data for the pellet. You can see down here the pellet. Okay, so I'm going to rescale this. It's got an absorbance unit of 300 at the base. It's made this steep pellet. So I want to change that to a scale of 2. And when I do that, you can see all of the data. Okay, here's the meniscus at 5.9. Here's the base. It's actually a little bit off scale at 7.2. I want to actually be able to process the data. So I want to process the data from here to maybe someplace like that. So there'll be this region over which we process. And then I don't necessarily want all of the data when I do the analysis. So what I want to do is I want to select scans. And so let's select 10 to 100. That's probably not enough. Let's go back. I've got to rescale. Oh, that can't be right. I wanted 10 and I wanted 100. I typed too fast. And now I'm going to go to 2. And so you can see it sedimenting. You can see the boundary starting to diffuse. You can see this radial dilution that happens. The plateau drops because the cell is sector shaped. Okay, I should probably make this closer to 80. Do it again. Okay, so that's okay. I could probably change a little bit more. Let's make this 15 and 90. Okay, so that's the data. And then I could actually analyze this data in a number of different ways. And so what I've done is I've chosen meniscus. Remember, everything corresponded to the meniscus. I've chosen what the base is because that's sort of the bottom of the cell. And so it won't go any further than that. But I'm really going to only collect or analyze data where it's cleared the meniscus, but it hasn't pelleted and I can still see something. So I'm going to write this data out. And then I'm going to analyze it by a different technique that I haven't even told you about yet, but it's one of the ways that we analyze data. And so here is the method, and boom, there's the peak. So this is a peak that is actually at around 5.8. Notice when I hold a cursor over this, it says 5.8. So this is the shape of the boundary. I'm actually analyzing data pretty late in the run. I could actually analyze data earlier in the run, and the boundary would be a little bit broader if I get rid of these data points. And you can see that the boundary actually spreads a little bit. Okay. And so this is one of the ways that I could analyze data like this. And the area under this curve gives me information about concentration. because it's absorbance. The peak position gives me information about the S value of the sample. But more than that, I can write that out. I can come back over here. And I can, actually what I want to do is cancel this. I want to come back in here. I want to create a new analysis from here. I want to say fit tubulin SV data. I want to use a monomer model because that's what I simulated. I know that its molecular weight is 100,000. I know its sedimentation coefficient is 5.8. I put in 0.27 for this. I know this is 1.44. I can pull in a data set. There's only one data set. That data set actually, we chose certain scans. Notice it remembers what I chose. And then I'll put in an estimate for the concentration. I think it's 1 mg per mil. Okay. So I can fit these. Blue means hold constant. When I click on it, it means fit. So I can fit these data to extract the S value. And so I'm now going to fit. And what it's doing is it's fitting these data to the Lamb equation, basically. Okay. It's showing two peaks, which is basically the raw data at different points in the cell, scan 15 all the way to scan 90. And it's showing you what the goodness of fit is. You can see that I'm getting something real close to 5.8. This is a chi-square. This is not actually an RMS. Let's go back here and do a root mean square. There's the data. I'm going to do a standard deviation. There's the S value. There's the concentration. You notice it's giving me really good results. When you fit computer data, you always get really good results. Real data is never quite this perfect. If I also fit the molecular weight at the same time, I should be able to do that. I'm going to say up here, S molecular weight, just so that I know this fit did something different. And now you're going to watch it down below, fit for the molecular weight and fit for the S value, fit for the concentration, and give me some kind of a standard deviation. Remember, I put in 0.005, but what you're doing in this presentation is subtracting one scan from another. And so that generates a square root of 2 in noise. And so this is bigger than 0.005 by the square root of 2. And so this is the technique that I can use for fitting these data. Notice I get 99.992 thousandths, 99,000, and I get 5.7997. I get the right S, I get the right molecular weight, I get the right concentration. Okay, this is how we actually solve data today. This is how I analyze data. Experimentally, I would not collect one sample. Experimentally, I would collect multiple samples at multiple concentrations, and then I would fit multiple samples. Notice I have other data slots here. So I would do multiple concentrations because what I want to know is there a concentration dependence? And then I could try to fit the real data to association, the real data for non-ideality. My models allow me to have virial coefficients, allow me to have other components, okay, allow me to do association reactions. Down here is a dimer model with non-ideality, a monomer, dimer, trimer model, etc. There's all kinds of models that I can create. I'm not going to show you everything about this. There's something called a model editor that allows us to create models that include more oligomers, that include non-ideality, that include all kinds of complexity. Okay, this is how we analyze data today. The modern sedimentation field solves the Lamb equation. And computationally, what you're seeing on the screen, when I was a student in the 70s, I would have to take this to a computer in a computer center, computers that were filled my office basically, in computer cards that I would have to feed into the machine and then come back tomorrow to get the results. We do all of that now on our modern computers, which are much faster, much more powerful than what I was used to using in the mid 70s. And then we can analyze all kinds of systems: velocity data, equilibrium data, absorbance data, interference data. I have a fluorescence system, so I do fluorescence data. I do fluorescence experiments in human serum, so I study antibodies in serum. And I can analyze the data in terms of the contaminants, in terms of non-ideality, in terms of self-association, et cetera. So the level of complexity is way, way past what I've introduced you to. None of this entirely makes sense unless you sort of understand what an S value is and a D value is, and sort of what the whole evolution is of hydrodynamic analysis. What's its size? What's its shape? Is it a monomer? Is it a oligomer? How big is the oligomer? What is the association constant for making an oligomer? Okay. So what we're going to do next week is look at a couple of papers from the 70s initially, the 1975 Friggin and Timoshev papers. I can honestly say to you, those are the papers that changed my life. Once I read those papers and understood those papers, I was on my way to being a scientist. I was on my way to thinking about science. The paper that I'm giving you that came from my lab 25 years later, 30 years later, was applying this type of technology, because we developed this software in the early 2000s for just these kinds of problems. Can we learn something about the mechanism of assembly that is way more complicated than what you would normally do from just graphical analysis? And so this is the technique that I have been using for my entire career, basically. And I have taken it to a level of detail that is, a lot of people think I'm a little bit crazy for trying to study serum and those kinds of systems, but it's where the field could and should be more broadly. Okay, so we're at the hour. So I'm going to stop sharing the screen. And I'm going to ask if there are any questions right now. Okay, so we just finished 4 hours on diffusion and sedimentation. I've given you a very sort of classical approach. And I've gone all the way back to the origins of the field. I've exposed you to some of the major breakthroughs that this technique has been associated with. It's been associated with a Nobel Prize, so that's the first thing. It discovered semi-conservative replication, and lots of other stuff. Okay. That would take me weeks to tell you all about. So there's an assignment out there. It's a couple of problems out of the Van Holt Johnson Ho book. It's still the graphical sort of analysis. It's sort of the straightforward analysis. I could have attempted to give you this software, but I knew it was going to be a problem because some of you are Mac users. So I elected to not do that this time. Apparently the 3 or 4 people I taught 2 years ago all were PC users, because it never came up. I know working with a Mac and a PC emulator, a DOS emulator, is a problem. It's hard to install and make work. And it costs money because you got to buy it. Okay, so we're back into that realm. All right, no questions. All right, we're meeting Friday at 12. I may be a little bit late depending on the faculty meeting, But basically, you and I are meeting at 12 to go over some of the questions, the assignments. And we're reading those 3 papers next Monday. Or next Tuesday, excuse me, a week from now. Okay.
Thank you,
Thanks,
Dr.
Dr. Correa.
Correa.
See you on Friday.
See you
Bye.
Friday.